Chromosomes in human beings. Sex chromosome and autosomes. Sex chromosomes. The chromosomes which determine the sex of an individual are known as sex chromosomes. Autosomes. The chromosomes which are responsible for determination of physical characteristics of an individual are known as autosomes. In case of human beings, there are total of 46 chromosomes which can be classified as 22 pairs of autosomes and 1 pair of sex chromosomes. Human males are heterozygous, that is, they have 22 pair of autosome and 1 heterozygous pair of sex chromosomes which are denoted as X and Y. Therefore human males will produce 2 types of gametes, viz. 22 plus X and 22 plus Y. Human females, however, are homozygous, that is, they have 22 pair of autosomes and 1 pair of sex chromosomes which are denoted as X and X. Therefore females in human beings produce only 1 type of gamete, that is 22 plus X. As we have discussed in the previous slide, during gamete formation, the normal diploid chromosome number is halved. This is called the haploid condition. All the eggs of a female have 22 plus X chromosomes. A male produces 2 types of sperms, one type bears the 22 plus X composition and the other, 22 plus Y. Therefore, in every 100 sperms, 50 have Y chromosomes and 50 have X chromosomes. Any one of the 2 types of sperms can fertilize the egg. If a Y-bearing sperm fertilizes the egg, the zygote has the 44 plus XY composition, and the resulting embryo grows to be a male individual. When an X-bearing sperm fertilizes the egg, the resulting zygote has the 44 plus XX composition. This embryo develops into a female individual. Therefore, sex is always determined by the other sex chromosome that they inherit from the father. Sometimes some of the traits are located on sex chromosomes, viz. X and Y. Sex chromosomes are also known as allosomes. Sex-linked inheritance is governed by certain generalizations which are Sons inherit sex-linked traits from mother and daughters chiefly inherit sex-linked traits from father. This type of inheritance is also known cross inheritance. Most of the sex-linked traits are recessive. Male offspring suffer from sex-linked disorder more often than female, as male's Y chromosome does not carry any corresponding allele. Females who are heterozygous, that is when the recessive gene is present on only one X chromosome, serve as carriers. Carriers carry the recessive gene. Since it is in heterozygous condition, it fails to express phenotypically. Females who are homozygous for the recessive gene are considered as diseased, as there is a phenotypic expression of the same. Males in single dosage of recessive genes on X chromosomes are diseased. There are no carriers in males. The examples of sex-linked inheritance are hemophilia, in which blood fails to clot due to genetically induced absence of an important clotting factor. Color blindness, in which the person fails to differentiate between red and green colors especially. However, color blindness for other color and color combinations have been reported. In the present slide it can be observed that in case of a normal individual, blood as soon as it comes in contact upon hemorrhage or rupture of blood vessel, it undergoes a series of reactions leading to clotting of the blood, which prevents blood loss. However, in case of persons suffering from hemophilia, blood fails to clot due to absence of clotting factor, and the person may die due to excessive blood loss, even due to small injury. One of the classical example is the Tsar family of Russia. One of the son of Tsar Nicholas was hemophiliac, thus he was get well guarded to prevent him from many injury. In case of color blindness, the sufferer is unable to differentiate between red and green color, as red and green color are often observed in the shades of matte brown. However, many other types of color blindness are observed. In the present slide there are some color blindness test circles which are also known as Ishihara circles. Inheritance of hemophilia and color blindness. From the previous discussion we can conclude that females with 20 combination of sex chromosomes are normal females. Females with XCX combination are called carrier females. Females with XCXC combination are called diseased females. Z are normal males and C are diseased males. Thus it can said that males are non-carriers for X-linked recessive genes. In condition 1, when a cross is made between a disease's father and a carrier mother, it is observed that daughters become carriers while the sons remain normal.
In condition 2 of carrier mother and diseased father, it can be observed that 50% of female offsprings are diseased. 50% of female offspring are carrier females. 50% of male offspring are diseased while 50% of male offspring are normal. Condition 3. Diseased mother and diseased father. Since all the X chromosomes carry the recessive gene for color blindness, therefore, all the offsprings are diseased. Condition 4. This condition refers to a situation where mother is diseased while father is normal. Here, it can be observed that all the female offspring are carriers while all the sons are diseased. The same type of X-linked inheritance pattern is also observed for hemophilia. In case of hemophilia, the recessive gene on X sex chromosome is denoted by H.
Y-linked inheritance is observed in case of male offspring only, as females do not receive Y chromosome from their father. Such type of recessive genes which are confined to Y sex chromosomes are also known as Hall-Andric genes. One such example of Y-linked inheritance is hypertrichosis, in which there is excessive growth of hair on pinna in case of some males.
The graphical representation of inheritance of a trait several generations of a family is known as a pedigree chart, and the study of the same can be done to forecast the expression in the future generations is known as pedigree analysis. Some of the symbols used in pedigree analysis are given alongside. It is recommended to learn these symbols to understand the pedigree chart, which can be used to analyze the expression of a particular trait in a family tree. In the pedigree chart it can be observed that a carrier female marries a normal male, which has been represented with a light blue square. In the first generation the expression is as follows. 50% of the sons were diseased. 50% of the sons were normal. 50% of the daughters were normal and 50% of the daughters were carriers. The diseased son of first generation marries a normal female and have 3 children, out of which 2 of the sons are normal and 1 is a carrier daughter. Similarly, 1 carrier daughter marries a normal male and out of her 3 children 1 is a normal male, 1 normal female and 1 becomes carrier. In this way we can observe the expression of the disease in future generations as well. Sudden or abrupt inheritable change in the genetic makeup of an individual is known as mutation. Characteristics of mutation. Mutation was first reported by Hugo de Vries in Evening Primrose Plants. Mutations are normally lethal but sometimes may be useful. Mutations are caused due to change in structure of genes, chromosome, or change in the number of chromosomes. Mutations may occur in nature due to various factors which are known as mutagens. Mutagens may be chemical substances like radionuclides, ionizing radiations and ultraviolet radiations of type C. Examples: sickle cell anemia. With sickle cell disease, an inherited group of disorders, red blood cells contort into a sickle shape. The cells die early, leaving a shortage of healthy red blood cells, sickle cell anemia, and can block blood flow causing pain, sickle cell crisis. Sheep with short hind legs in Australia.